just would like to share with you some thoughts on the design of experiments, especially of omics experiments, which on one hand are a new challenge, but they are also on another hand, it's possible that they suffer from old caveats of design of experiments. Okay, so let me see. Yes, I think you should see outline now. So I will try to look first at the common features of the omics technologies, which make them especially interesting from design point of view. And I will look at the issues of bias and variability, which are very close linked to the omics technologies. I will take a look at the design of experiments issues and some techniques which are relevant for design of experiments. And we will look at a couple of examples. One of them will be development of class prediction procedures and the designs design features which are important there. We'll look at the analysis of labeled mass spectrometer experiments, and I will finish with some hopefully simple conclusions. So what is omics? Now, omics is an explosion of different in different areas. We have a lot of omics in genomics, cognitive genomics, comparative genomics, and so on. We have proteomics, of course. You have metabolomics, and you have the name omics appearing almost everywhere, also in Asterix and Obelix. Now, all of these techniques have a lot of common. First, they pose special problems, but the aims of omics experiments can be generalized into can be categorized in three main classes. First, you have class discovery. For instance, you may want to come up with a gene or protein signatures to find new disease subtypes, some kind of molecular subtype of breast cancer, for instance. You may want to look at class comparison. So it means that you may want to, for instance, compare protein abundance between biological conditions, and in this way you could perhaps see whether some proteins are more abundant in sick than in healthy people, and this could give an idea of what might be the mechanism of the disease, for instance. And finally, you can have class prediction, where you could use gene or protein signatures for diagnostic purposes, meaning that you might want to, for instance, decide upon a signature whether someone is sick or healthy, or whether someone will develop disease progression in 5 years or not. So these are the three different types of omics, of goals of omics experiments, and depending on the type, we may need to adapt the design to the particular goals of the experiment. Now, omics technologies are sophisticated and impressive because they are based on advanced scientific principles. They use complex instrumentation, and they produce massive amounts of data. This is commonly known as high throughput. Now, these are, of course, very powerful features, and they, as such, are constituting a big advantage of the omics technologies. However, at the same time, these features make the technologies vulnerable. And why is that? First of all, these techniques are highly sensitive. You can have systematic effects due to time, place, reagents, personnel, to many different factors, and this means that reproducibility of the results obtained by using an omic technology may be actually impaired. Also, you can have considerable variability because the complexity of equipment, for instance, means that there can be quite a bit of a random noise which is present in the measurements which you obtain. And finally, it's also because of these reasons that naive data analysis can lead to erroneous conclusions, because if you, for instance, do not adjust for the systematic effects, you may actually find some differences which are just simply differences due to some systematic effects rather than the real differences between, for instance, two biological conditions. So because of this, one might say that the analysis and design of experiments which involve omics technologies need to be let's say, carefully considered. Unfortunately, this is not often the case, and I will use this case study as an example. This may be very well known to many of you. This is a case study in proteomics. It involves use of mass spectra to discriminate between ovarian cancer and normal samples. There was a series of 4 papers. The original paper by Petricon et al. Was published in Lancet, and of course it made a big impact, and it was very it was very well commented and very well present in the discussions. Then there was a paper by Conrads, by the same group, which actually looked at these data in a little bit more detail. And later on, Keith Baggerly and his group, these are bioinformatician statisticians for MD Anderson Center in the US, they looked at these data and they discovered some issues with the results which were published in these two subject matter papers. And also, Soras and Jian looked at the data in more detail and commented upon the reliability of the results. Okay, so what was the message which was published in Lancet by Petrikon and al.? They took 100 ovarian cancer patients, 100 normal controls, and 16 patients with benign disease, this 216 in total, and they developed a classifier. So they took 50 cancer and 50 normal spectra, and they trained a classifier. And then they took the classifier and they trained the algorithm on the remaining 116 samples, all right? So the results were really impressive. They correctly classified 50 out of 50 test ovarian cancer cases, and they correctly classified 63 out of 66 of the non-malignant cases. So they had 100% sensitivity and 95% specificity. This is like you cannot be better than that in medicine, actually. So of course this paper made a lot of noise, let's say, and this paper was pointing to the direction of mass spectrometry as a potentially, a perfect diagnostic tool for ovarian cancer, at least. Okay. Now, Conrads and colleagues, and you can see Petrikon also as a co-author of this paper, they look at this data a little bit in more detail. So they took these samples and they actually processed them with a higher resolution instrument. So they use the QQ-TOF machine. They especially paid attention to the quality assurance and quality control, and this is commendable because thanks to this feature of their publication, we can actually take a closer look at these results. And with these techniques and with this processing, they actually improved the results which were shown in Lancet. Essentially, they found that they had achieved 100% sensitivity and 100% specificity for identifying cancer from normal. Now, this is, of course, you cannot do better than that. 100% is the maximum, okay? Now, what happens is that essentially, well, you see here two spectra. You can say that one spectrum is from the healthy individual and the other one is from the cancer individual. You have here spectrum for the normal and the spectrum for the cancerous sample. And if you zoom in, perhaps in this region around like 8,500 and to Z, so Daltons perhaps, you see that, for instance, in this sick person sample, you see a peak which is not there at 8,600 for the healthy individual, and likewise you see a peak around 7,050 for the sick spectrum, and you don't see this peak in this healthy spectrum. So using features like this, you could actually try to distinguish spectra for normal samples from spectra from disease samples. Okay, so it looks that there is a tool to discriminate between the different types of samples. So Keith Baggerly, Jeff Morris, and Kevin Coombs from MD Anderson took a look at this data, and we, as I said, the group of Conrads and Petrikon is to be commended because they actually made the data available for the other researchers. So it was possible Keith Baggerly and colleagues could take the original Petrikon samples and they could take the data for the original samples, and they also had a new set of 253 spectra, 99 normal and 91 normal and 162 cancer samples run on another platform, and they could actually take a look at this data more closely. And what they did is, remember, the previous results were based on these 216 samples, okay? So what you could do is you could take the classifier developed on these 216 spectra and apply it to this new set of 253 spectra and check whether the predictions or whether you are able to distinguish the normal and cancer samples. So this is these are the results which Baggerly and colleagues published. So if you look at the data set 3, so this new set of 253 spectra, you can see that there is a clear separation there. So you can see that if you use features from this data set, you can clearly see that, you know, these are like spectra, spectrum number. So you can see that there's a group of spectra which is similar in their features and which differs from the group or the other group. So it's like the difference between normal and cancer samples are seen there. If you use the features of the spectra from these data. Now, if you use the classifier developed from dataset 2, so developed by Patrick and colleagues, you see this picture which is at the bottom of this plot, and this basically means that there's absolutely no distinguishing between any samples. There's no grouping which you could see up, in the upper panel. You don't see any reasonable classification or discrimination in the lower panel. So this means that this classifier, which was supposedly leading to 100% sensitivity and specificity in ovarian cancer, it doesn't work on dataset 3. But obviously, we would expect that the classifier used for diagnostic purposes should work on an independent dataset as well. So something went wrong, and the question is what? So we will come back to this issue, but before looking at that, let us go back and look at the goals of the design of experiments, okay? So there are a couple of goals which the proper design of an experiment should actually achieve. First of all, we want to prevent the bias, all right? And the bias is a systematic average difference between the estimated and true value of a parameter. So in other words, you really want to avoid the situation when, for instance, you estimate that there is a difference between two conditions, whereas there's no difference, okay? You would like to maximize the precision of your inference, okay? So you would like to minimize the variability of the estimates, for instance. So it means that you would like to be as precise as possible in the statements regarding a particular parameter. I don't know, mean blood pressure or mean abundance of a peptide or protein. And then you would like to control the probability of error, especially in the context of testing. So you would like, for instance, to control the probability that you will reject the null hypothesis of no difference. If there is no difference, you would like to reject the control probability of this false positive finding to, say, 5%, okay? So proper design of experiments should allow you to sort of like address these issues. Now, we can visualize this in this sort of like symbolic representation. At the lower right side, you see the perfect situation. You have very low bias because you are on the target, and you have very low variability, so it means that you have a very precise estimate of the quantity you are aiming at. On the right-hand side, in the top panel, you see the situation when there's low bias, because on average you are sort of like, if you would average the points, you would hit the target, but there's high variability because the points are really spread across the whole target. If you look to the left side, to the upper panel, you see that we have a very low variability in panel A, but we are really off the middle of the target, so you clearly see there's a bias. And in the panel C, in the lower, let's say, left-hand side corner, you see that you are not only having a high variability, but you are also off the center of the target. So essentially, you are in the worst situation as possible. So ideally, we would like to position ourselves in the situation D, and we would like to avoid the other situations. Perhaps situation B is not that bad, but we would have bad precision of our statements, but certainly A and C might be problematic. Okay. Now, if we talk about this variation, all right, so how could we, what kind of variation we have to deal with? Okay, there are three basic types of variability which can affect measurements in our experiment. First of all, you have technical variability. This is related to repeated measures of the same sample. And this may be due to the biological sample processing, due to the equipment, due to the operator, and things like that. So it essentially means that if you measure the same sample 3 or 4 times, these 3 or 4 measurements may differ from each other, and this is technical variability, and they may differ a lot, and this is a problem because this means that you have a lot of noise in your measurements, or they can differ very little, and that would be good. Then you have biological variability, which means that the measures of different biological samples differ. And this is something which we have to live with, and actually this is what we'll encounter if we go to the population of people and to measure the blood pressure. This blood pressure will vary among the people, right? And this is the biological variability, and this is something which will be present in the measures always. And we can have something which is institutional variability, which is related to the use of the same technology in different labs. And obviously this is due to differences in equipment, in the protocols which are applied in different labs, in biological populations which are available in these labs, and so on, right? So in a way, institutional variability is on top of this technical and biological, and it adds additional factors which may influence the precision of the measurements, okay? So now, to minimize these different types to sort of like address different issues which I just mentioned, like variation and bias, we have a couple of techniques which can be used in design of experiments. Now, minimization of variability or increasing of the precision is obtained by using replication or blocking. So these are the two techniques which allow you to address the issue of increasing the precision of your experiment. On the other hand, to protect the bias, you can use random sampling and randomization. And we will talk about these different techniques in turn now, okay? So replication means essentially that you make repeated measures of something. So you can have technical replication where you take repeated measures of the same biological sample. When it's useful? Well, it is useful if you evaluate an assay, right? So if you want to assess the reproducibility of the assay, right, then because you would like to, as I said, you would like to have the same measurement for the same, well, you would like the repeated measurements for the same biological sample to be as similar as possible. So you would like to have a small variation there. So technical replication is useful if you want to assess how stable are the measurements from your equipment. And actually, for instance, it is well known that these RNA-seq RNA sequencing techniques, like using these new next generation sequencing technologies, they have a very low technical variability, which means that they are very precise. So essentially it means that if you want to have an idea of what is the expression level for a particular sample, you can just do one measurement and more or less you will be fine in terms of the estimation of the true level of the expression in a particular sample. Obviously you can have biological replicates. And this is important if you want to generalize your results to the population, right? Like in class prediction or case, for instance. Why? Because you want to, in your experiment, you want to see this biological variation because you want to know how this variation may affect your prediction, for instance, okay? And this means that in your experiment, you might need to include biological replicates to have this possibility to generalize the results, but also by taking several biological replicates, you are able to if you average them, you are able to minimize, well, to sort of like reduce the influence of the biological variability on your estimates. And obviously, institutionally, you can, if you have institutional variation, you can take multi-institutional experiments, and this is especially useful if you look at, for instance, development of diagnostic assays, because you would clearly want to check whether your diagnostic assay performs the same way from one institution to another. And this essentially means that you should be able to assess this in your experiment, but also again, if you take institutional replicates and sort of like average the performance across the replicates, you will be sort of like getting a better assessment of your misclassification rate, for instance, taking into account the fact that you will, if you will be doing your diagnosis in different labs, there will be also differences between the labs. So replications allows you to get a grip on the variation and variability, and by taking replicates and averaging them, you can actually make the, well, increase the precision of your estimation, for instance. Generally, it is better to have more biological replicates than technical ones, and the reason is, as I said, first of all, generalizability. The results of your study may be more generalizable if you have biological replicates, but also biological replicates necessarily carry with themselves also technical replication, because you have to measure the biological samples, right? So it means that by making more biological replicates, you also take into, well, you also carry with, they carry with themselves technical replication, technical variability, and you also address this issue by taking replicates at the biological level. Now, what blocking means? Now, blocking is used when variability between blocks is larger than within blocks, okay? And an example of block is a microarray, for instance, or a spectrum. Essentially, it means that you have like you can have like a group of measurements which are similar, but if you have another group of similar measurements, it will be very much different. And this means that if you start to look across the blocks, the variability will pop into your measurements. Okay. So the idea is to compare samples or interventions within the blocks, so that the comparison of the conditions or comparison of samples is not done across the blocks, so it's not subject to the between block variability, but it's done within the block, so then you can actually remove the between block variability from the comparison of your samples or your conditions, right? And an example in mass spectrometry, for instance, are labeling techniques like iTrack or, you know, O16, O18 labeling, SILAC. So we know that there is considerable variability between the spectra in mass spectrometry. So comparison of samples within the spectrum removes this variability. And this means that then essentially you can compare two samples. If you use O16, O18, you can compare two samples within the same spectrum. So then the between spectra variability will be removed from this comparison. And therefore the precision of your estimates, like for instance of the relative abundance, will increase. Now, what about random sampling? Random sampling means that you randomly select samples to be included in the experiment. This is aimed at prevention of the selection bias, and the selection bias is bias due to inclusion of samples which are non-representative for the population, which basically means that if you have non-representative samples and then you try to generalize your results to the population, you will make an error, you will have a bias, because the samples will not describe the population, will not be representative of the whole mixture of subjects in the population. So it is important to use random sampling from the point of view of generalization of the results. Unfortunately, most often people use convenience sampling. They just use available samples which are there. So for instance, if you run a lab in a which is associated with the hospital, you take the data from which is available in the hospital, and obviously the people or patients who come to the hospital may not be representative for the whole population of the people, but they might not even be representative for the population of the patients with a particular disease, because if you think about like clinical hospital, you may expect that patients coming to a clinical hospital may be more advanced in their disease than, for instance, patients coming to a small hospital somewhere in a community. So this is an issue, and this has to be carefully thought about when you run experiment, how far you can generalize the results of your experiment, and what sort of bias might be in your results given the fact that you had the subjects available which you had. Now, randomization is something else. Randomization means that you randomly assign intervention to samples in the experiment. And the main idea is that it allows you to balance samples characteristics across interventions in such a way that the observed difference can be attributed only to the effect of the intervention, right? So this means that you avoid the bias in comparison in the sense that, for instance, if you would like to compare the results of treatment of two groups of patients, and one group would be older than the other, then if these two groups were treated by different, using different drugs, then if you compare the groups as such, you will be comparing not only the effect of the treatment, but also the effect of the difference in the age, right? And then the problem starts that the problem pops up that if you have observed the difference, well, is it due to the intervention, to the treatment, or is it due to the fact that the one group was older than the other? And randomization allows you to avoid this issue because then by random assignment patient treatments to the patients, essentially you would balance the distribution of age between the two groups. Of course, this is possible to be done only if you can randomize the intervention. So there is a limitation to the use of randomization. Okay, so from point of view of designing of the experiments, we have basically 4 techniques: replication, blocking, random sampling, and randomization. So let us see how these techniques might be put into practice. Now, let us revisit this example of Petricon. All right. So we have these data, and as we saw, by using the classifier developed by Petricon and/or Conrad, you cannot really discriminate between the normal and cancerous samples in the third data set, which was available to Baggerly and colleagues. Okay. So what has happened? Now let us look at this paper by Conrad, where, as I said, they were looking at the quality assurance there. Now, if we can look at their figures 6 and 7 there, all right? Now this is the sample number is on the x-axis, and you have record count, so let's say the total ion count on the y-axis. And what you can see is that there's a clear difference for the samples which are larger sample number. And actually what happens is that on day 1 and on day 2, the record count was about 3.5 times 10 to the 5, so it was more or less the same. So the quality of the equipment was more or less the same. But now you can see that on day 3 something happened. All of a sudden you started to see spectra which had a smaller record count, so something happened. I don't know, maybe the protocol applied to the machinery was changed, or maybe something happened in the equipment setting. Now they themselves indicated that there were 32 spectra that were lesser quality, and they were generated at the end of experimental run, suggesting that a deviation in the process had occurred. So what happened in this experiment is that on day 3, they had problems with the quality. Okay? Now, this is not a problem. This happens, of course. But now we look at the same picture, record count on the y-axis, sample number on the x-axis, and now we look at which samples were processed. And what you can see is, from their picture, is that cancer samples were processed mainly on day 1. Control samples, however, were processed on days 2 and 3. But there were quality problems occurring on day 3. So the question is, what do we discriminate between? I mean, do we discriminate between the control samples and cancer samples, or do we discriminate between the good quality spectra, which were seen on day 1 and day 2, and bad quality spectra, which were seen on day 3? And this is actually unfortunate happening in this experiment. And this is the classical issue related to so-called confounding. Confounding means that the association between the response and the factor is actually influenced by the third factor, which is associated both with the response and the factor we are interested in. So what happened here is we have mass spectrometry intensity measurement, so our response. We have the sample status, so cancer control. This is let's say, our factor we are interested in. But we have a day of measurement, a third player, which comes in between the two. And the problem is that by chance, there has been an association between the day of measurement and intensity measurements and the response. And the association is that on day 3, intensity measurements were of lower quality than for the first 2 days. In the same experiment, however, we had also association created by the study design between the cancer and control samples and the day of measurement, because on day 1, cancer samples were processed, and on day 2 and 3, only control samples were processed. So essentially what you see is that you have, if you look at the distribution of the day between the cancer samples, you have 100% of day 1 among cancer samples, but 50% of day 2 and day 3 and 0% of day 1 in the control group. So there is an association which was unfortunately created by design, by the decision to run the samples, in an ordered way. And you have an association between about the intensity measurements and day of measurement, which happened by chance. But because of this, there's an observed association between the intensity measurements and sample status, which is induced by these two associations. And because of this, if you forget about the day of measurement, essentially what you see is that for some reason cancer and control samples are related with different intensity measurements, whereas this effect is actually due to the fact that there was an association between the day of measurement, between the sample status, and the association created by chance between intensity measurements and the day. So this is actually a design problem. And one could ask, hey, how could you prevent that? Well, very simply. You could prevent it by randomizing the order of processing the samples. In other ways, you could assign the measurement days, which would be sort of like your intervention. You could assign them randomly to each of the samples. So you could, with equal probability, for instance, so you could, for each sample, decide, okay, I will analyze this sample on day 1, 2, or 3, right? And the probability of 1 to 3 is 1/3. And this means that by using such a random processing of the samples, you would balance the distribution of the days within the groups. So day 3 would still be having worse quality of the MS measurements, but these bad quality spectra would be distributed evenly between the cancer and control samples, and this means that there would be no difference in this respect between these two types of samples, and this would not influence the classifier to the extent which we saw in the original results. So the main problem with this design was that there was a let's say, created by the design, there was this link between the day and between the sample status, and this actually resulted in a spurious association, which we observed in the results. And this is something you often see, and I've seen this several times, that, for instance, there's a very pronounced day or temporal effect. For instance, from week to week, MS measurements change, and that's actually due to maintenance, for instance, of your machinery, or the fact that some people start to run their experiments and they fiddle with the equipment settings of the equipment, and later on they are not put back to the same status as they were in the previous week. So temporal effects can be expected in omics technologies, and therefore the order of processing of the samples across the time should be carefully considered, and therefore randomization should be used to prevent the skewed distribution of the processing days or temporal effects across compared samples. Now, if you look at, as I said, Soras and Jean looked at these data, and they reanalyzed this third data set on which the problem with the classifier was discovered, and then actually found perfect classification rules using only 2 MZ features. So actually what you can see here is that using, let's say, value of the intensity measurement at the mass to charge value of 245 and about 2.8, actually you see that there is a perfect classification possible using like a straight line in this plane, right? However, if you know a little bit about the mass spectrometry, I mean, these 2 coordinates lie really in the region where a lot of noise is happening. So essentially, what Soras and Jean say is that the ability to discriminate between these samples based on the mass to charge values of 2.79 and 245 reveals the presence of significant experimental bias not related to disease pathology, that likely involves machine noise. So it seems that there also they point out to the fact that there were some issues with the experimental design regarding the contents of the spectra, and essentially they say that investigators in this field may minimize their chances of false discovery by careful experimental design and by using routine statistical methods. So essentially, they point to the fact that simply randomizing the order of the samples might actually alleviate at least of some biases which we see. So this was pretty striking example. And if we talk about class prediction, about discrimination, you can say that this is really a rigorous process. You should start from, like, say, signature identification or whatever, a classifier identification, and then you should try to have internal validation. So validation using the data you perhaps where you identify your signature. And this was actually done by Petrikon and others, because you can for instance, by internal validation means that you could split your data into two halves. On one half you develop your classifier, on the second half you test your classifier. But then there should be external validation. So this means that you should really take a sample which is external to the one which you used internally to develop signature and validate it, and you should check whether your classifier works fine. And in this respect, this failed in this example, because if you took this third data set, you could see that using this third data set, you couldn't confirm the results which you saw initially. All right. One thing which you should beware there is that if you increase the complexity of your classifier, you obviously will be doing better when you will be developing, assessing the classifier performance of the data set which you use for training or your classifier. So this is a plot taken from the paper by Allison. You have this predictive accuracy on the y-axis. You have model complexity in the sense of number of parameters or number of features which are in the classifier. And you can see that if you make the model more complex, on the training data set, you will be achieving the better predictive accuracy. So essentially, you can almost like, at one point, you might almost completely predict your data. However, if you look then at the performance of this classifier in a test data set, you can see that by making the classifier more complex, you can only achieve improvement only to a particular point, but later on, you will start to lose. And why? This is because you start to overfit. This means that if your model becomes too complex, it starts to look into the features which are very specific for the training data set, and on the test data set, this feature will not be present, so therefore the classifier will start to make errors if you apply it to external data set. And this is a common problem, because it means that this actually means that if you make a very complex classifier for your data, the classifier is good for your data, but it will not be good for data from another lab. So this is a very, very subtle point where you need to be careful, and there are statistical methods which allow you to sort of like penalize you for using too many features in your classifier. Now, as I said, external validation is important, and perhaps this was one of the things which Petrikon's group didn't look into too carefully. And external validation means that an independent study is needed to confirm the results of a previous study in order to reduce the play of chance and the potential for biases. Well, what are the criteria for external validation? You should pre-specify the experiment. So you should completely pre-specify a list of features along with a completely pre-specified decision rule to classify new subject. There should be independence, so new subjects should be recruited either prospectively or retrospectively, but they should come, and there is homogeneity, they should come from the same or a similar population on which the classifier was developed, right? So you should really not fiddle with the classifier. You should take independent data set from the population you used in your previous exercise and then check how the classifier works. Unfortunately, there are common mistakes which go against these rules, like a mistake which you often see, and these are the actually mistakes which are listed after the papers which looked at the microarray examples, is to include part of the training sample in the validation study, or to include other types of subjects in the validation study, or use another measurement techniques, for instance, to swap RT-PCR for microarray or vice versa. Or perhaps to adapt your prediction rule, to adapt your classifier to the new sample through changing the list of features, or to change a little bit the prediction rule, or the cutoff which you use to decide whether you have like sick or healthy people, or stuff like this. And one might say that this seems to be like pretty obvious mistake, but you can see this happening over and over again. And this is an example of a famous gene signature in breast cancer. New England Journal of Medicine, very well, one of the top journals in medicine. There was a gene expression signature developed for a prediction of survival in breast cancer. Essentially, what happened is that they developed a signature based on about 70 genes, and you can see here the probability of remaining metastasis-free as a function of years for the group with a good signature in blue and poor signature in red, and clearly there's huge difference. And essentially, this indicates that based on the signature, you could predict how whether someone would remain metastasis free or not within, let's say, for instance, 6 years or 5 years. Okay, fine. The question is, how did they validate this? Well, they did use a study with 295 individuals in the validation set, but they actually included in this data set 61 individuals who were used in the training data set. So actually 1/5 of the validation data set was the training data set individuals. So obviously this skews the evaluation of the performance of the signature towards the results seen in the training data set. And the Ransohoff actually says that the degree of discrimination shown might have been inflated by overfitting, because the validation data set looked similar to the training data set, right? So if we look at this example, this signature is well known. It is used clinically, but if you look at its the process of development of the signature, you could see that the same error which I mentioned, that the independent validation was not completely independent. This error has been made. Now, finally, let me go into the issue of the analysis a bit. Okay. Now, I will consider the case of this proteolytic 18O labeling case. Basically, this is the case, as I told you, of applying blocking into your experiment. So let's say that you have two conditions, condition 1 and condition 2, and you have a protein mixture in two conditions. You digest it to peptides, okay, and you want to compare these conditions. So what you can do is you can actually put the conditions 2 into heavy oxygen water and allow the O16 oxygens to be replaced by O18O oxygens, which will make the molecules heavier by 4 Daltons. So essentially, what you would get after the exchange of the oxygen atoms, that you would have O18 atoms attached to the molecule from the condition 2, and because of this, this molecule would become heavier. And then it means that you could put the mixture of the two conditions peptides into your mass spectrometer. And what you would expect is that for the molecules from the second condition, you would observe these peaks shifted as compared to the molecule from the first condition by, well, let's say if we talk about charge 1, by 4 Daltons. So it means that you could look at the two sets of peaks, and essentially it would mean that the peaks which follow 4 Daltons, after the first peaks, you would say, okay, this is the condition 2. So what you could see, what you could do is you could take the ratio of the 5th and the first peak in such a cluster of the peaks, and this would be a measure of your relative abundance between condition 2 and condition 1 in terms of the and that would mean that you could have an estimate of the relative abundance for condition 2 as compared to condition 1. Now, obviously, you already see here that it's a biased measure. Why? Because you can see that there is a little bit of the input of the unlabeled sample, unlabeled molecules from condition 1 entering the 5th, 6th, and 7th, and so on peaks, which are belonging to, predominantly in this example, to condition 2. So this means that the part of the 5th peak is actually due to the unlabeled sample, and therefore this ratio of the 5th to the first peak is a biased measure of the relative abundance. Now, this means that this is naive analysis would be actually making the results problematic. There's another issue there. There is something which is called a heavy oxygen water impurity. You have O17 atoms in heavy water. So essentially, it means that you can have like 95% of your heavy water O18, 1% O16, and you can have even like, for instance, 4% of O16. Which essentially means that you will not get all the labeled samples heavier by 4 Daltons, but they can actually be heavier by 0 Daltons if there's no exchange, because you have a couple of O16 Dalton atoms exchange with those which were in the molecule, or you can have a shift by only 1 Dalton when only one O16 atom is replaced by O17, or you can have shift by 2 or 3 or 4 Daltons. 4 Daltons would be what we want, but because of the water impurities, you will be having shifts of the condition 2 peaks by even 1 or 2 or 3 Daltons. Now this means that you have a complex mixture of these 6 isotopic distributions, so of these sets of peaks for condition 2 over the condition 1 peaks. And this means that this the analysis of these results from such an overlapping distributions is very much complicated. What you need to do is you need to have a model which recognizes the fact that there is a overlapping of these different distributions, which corrects the relative abundance measures, which would be naively taken as the 5th peak to the 1st peak. Or you can use a suitable experimental design, something which is called label swap, which basically means that in one mass spectrum you should label condition 2 with O18, but in another mass spectrum you should reverse this, so condition 1 should be labeled with O18. And in this respect, you could sort of like correct, you could, by using appropriate techniques, statistical techniques, you could correct for this overlap, and you could get an unbiased measure of the relative abundance of condition 1 to condition 2, or vice versa, of the abundance of a particular peptide in condition 2 sample as compared to condition 1 sample. So this is an example of a situation where by blocking you could reduce the variation of, well, you could increase the precision of the comparison of two conditions, but you need to take care about potential bias, which is due in this particular case to this overlap of the peaks. So the obvious conclusion is that omics is not an exception, and all rules of design of experiment apply. Essentially, it means that you should take care about bias and precision of your inference, and you should try to use like randomization, random sampling, blocking, replication to try to minimize the potential spurious effects of systematic effects or increasing the precision of your experiment. So you should plan and analyze your experiment carefully, and in this respect, the omics experiments are no different from the experiments which were run in the past and use these classical rules of design. And maybe even a more obvious conclusion is this quote which I took from Ronald Fisher, who is the father of applied statistics, who said, "To consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of, but he cannot rescue the experiment anymore."